Hi everyone, so we're going to move on to our next system here, our muscular system. So as we always start with, we're going to talk about some different functions that we can see with our systems here. So first function here is going to be movement. So that's number one. So movement can be locomotion, so that's going to be with our voluntary skeletal muscle, cardiac action, so our heart constantly pumping our blood through our body. That's our involuntary cardiac muscle. And then visceral peristalsis, that's the movement of our intestines and moving all of that food through our body. And that's going to happen with our involuntary smooth muscle here. Our next function here is the generation of heat. So every time that our muscles contract in our body, we are creating some heat from that. So that's going to help maintain that consistent body temperature, which is going to be really important. So that's why if you're really cold, you're going to start shivering. Those are little muscle contractions that are going to help generate heat to keep you warm. And our last function here is going to be energy storage. So any of that excess glucose is going to be stored in as glycogen in our liver and in our muscles. So we use our muscles as storage units for all of this energy that we get from glucose. So saved as glycogen. Okay, so let's take a look at some of our structures here. So we have some connective tissue coverings that we're going to talk about here. So our fascia is a connective tissue covering that surrounds each muscle, and it attaches to our skin. Tendons, we know, are going to connect our muscle to our bone. And then aporoses are going to connect muscles to other muscles. So different things that we're going to see here. Fascia connects muscle kind of to our skin. Our tendons connect muscle to bone. And then our aporoses connects our muscles to muscles. So let's take a look at these. So we can see our skeletal muscles connecting to our bone with our tendons here. And then all this white in between here, that's our connective tissue. These are that connection between muscles and muscles here. So more covering. So when we're talking now even smaller into our actual muscle cells. So our epimysium is the outermost covering. And we go down to our inner covering here. So our epimysium is a layer that closely surrounds that skeletal muscle. So it's around the entire skeletal muscle. Our perimysium is going to surround our fascicles. So we're going to define that term in just a second. And then our endomysium is going to surround each muscle fiber. So going from the outermost, our entire muscle, to our fascicles, to our inner muscle fiber here with these different connective coverings. So if we take a look, our fascia is all the way on the outside. Our epimysium is covering the muscle around here. Our perimysium is covering each of these little groupings that we can see in here. That's our perimysium. And then if we get even smaller, our endomysium is covering each one of our fibers here that we can see. So this is an individual muscle fiber right here. So each fiber is going to be covered by our endomysium. And if we also take a look at how this is broken down, so we can see this is our entire muscle. So this is our muscle. We can see our fascicle is one of these bundles in here. And as we go even smaller, one of the bundles that we find within our fascicles is a muscle fiber, those muscle cells that we saw under the microscope. And even further, one of those connections are called myofibrils. And if you go even smaller, we see all these filaments. And those filaments is really where those action comes in with those different cells. So let's talk about skeletal muscle fibers, going down to our smallest unit. So our skeletal muscle fiber is that single muscle cell. Our sarcolemma is the membrane covering each cell. Our sarcoplasm, think of that ending, plasm, is that cytoplasm that's within that muscle cell. And then these myofibrils are going to be this really important structure here. So it's two kind of protein filaments, actin and myosin. So those myofibrils are going to be really important. We also see our sarcoplasmic reticulum. So these are different channels that surround each myofibril. And then we also see transverse tubules, or sometimes you'll see it written as T-tubules. And those are, again, channels that run perpendicular to our sarcoplasmic reticulum. So if our sarcoplasmic reticulum runs this way, this is our SR, our T-tubules are going to run this way. So if we take a little bit of a look here, these nice little our yellow tubes right here, these are our T-tubules right here. We can see each of our individual muscle cells in each one. Our myofibrils we'll see a little bit closer, but again, we'll see this nice sarcoplasmic reticulum running the opposite direction of our T-tubules. Sarcoplasm, that nice cytoplasm in the cell, and then our sarcolemma, which is that membrane around. So our striation patterns is really going to define those microfibrils here. So we have our I bands to start, which is our light bands, and you'll see why in a second, why they're called that. And these are composed of our thin actin fibers. And these are going to be attached to a structure we call the Z-line. So a sarcomere, which we're going to see that definition when we go over this a little bit in more deeper context, is a distance from one Z-line to the next. So if we have a Z-line over here, and we have a Z-line over here, this distance is what we call the sarcomere. Then we have A bands. These A bands are our dark bands, and they're composed of thick myosin. So they're dark, we have thick filaments here. And sometimes they're going to overlap with our actin, which is why they're going to appear darker. There's more stuff there. So the area known as the H zone is with myosin fibers only, and our M line. So we're going to define all these terms in just a second with our picture. So myosin filaments are attached to the Z-line by a protein called titin. So let's take a look at these images here. So if we look here, these filaments in between these Z-lines, so this is our Z-line right here, going all the way down. The ones that are in between, not really connected to anything, those are our myosin filaments. And our thin ones are going to be connected to this Z-line right here. So this entire thing is our myofibril. Getting even smaller into these proteins here is our thin lines with actin, and our thick, the middle lines with our myosin. So if we're looking at just our pictures here, we can see that our H zone, this is where we're just going to see our myosin filaments. Our A band is our dark zone, where we see our overlap with our myosin and our actin. Our I band is where we're going to see just our light band here, is going to see just where we see those actin molecules. And then remember, from Z-line to Z-line, that is the size of our sarcomere. So a little bit closer here, we're seeing our thin filaments. They're connected to our Z-line over here. So our thin filaments, again, are called our actin. And then we see our thick filaments that are kind of in between. Those are our myosin. We see our Z-line come down. And we see this overlap here with our A band. So let's bring this down. Nice overlap here. So the overlap between our A band and our actin and our myosin is what makes up that A band, which is going to be important when we talk about the actual contraction of muscles. But it's important to realize all of these different components before we even get to that contraction portion here. So there's also two other proteins here that are going to be really important when it comes to muscle contractions. So when the muscle is at rest, meaning it is not contracting, our proteins called tropomyosin and troponin are going to be blocking the binding sites on our actin molecules. This is going to prevent any linkages from being formed or any connections being made between our actin and our myosin here. So when these are blocked, we'll see our actin proteins. We'll have that filament. We'll also see these tropomyosin and troponin blocking these little sites that we have on that molecule. But when we're in contraction and calcium ions are present, because we need calcium for contractions, we're going to see that our troponin is going to change shape, kind of like an enzyme. And as a result, the position of our tropomyosin is going to be affected, and we're exposing those binding sites on our actin. And because those binding sites are open, our myosin filaments are going to be able to attach to our actin molecules here. So if we take a look, this right here, our thin filament, this is our actin. We can see our troponin and tropomyosin over here, blocking those sites. Right here, this is our thick filament, this is our myosin. So once those filaments move off those binding sites, these little heads here of our myosin are going to be able to attach to the actin molecule to create that muscle contraction, so we can actually move the muscles in our body. So that is where we're going to end for today. If you have any questions on just these terms so far and being able to identify those, please ask. These are going to be really, really important terms that we need to continue to use throughout our muscular system here.